Okay, let's get started. This open for discussion. But before we start asking questions, I want to introduce two very important persons, which are the head of Transgenesis at NIMR, Sarah Johnson, and at LRI, Ian Roswell. They will be co-head of Transgenesis at the Creek, so basically from 2 weeks' time, we'll have two heads of Transgenesis. So it will be nice for them to show us what they can provide when we'll be at the Creek. And then we'll open the floor for discussion, and please ask question, any kind of question, to the expert that will introduce themselves in due time. So, but for now, Sarah.
So we've divided this quick introduction to our service into two bits. I'm going to quickly show you what we're doing with CRISPR across the two institutes, and then Sarah's going to talk about the vision that we have for what's going to be termed the genetic manipulation service. Just talking about what we've done over the last year at the LRI to establish this technology. We aim to create this platform, a CRISPR-Cas platform, very much with the idea we want to take forward to the Francis Crick Institute, where we've got a sequence to mouse service, and we can. That doesn't mean that every project has to be from sequence to mouse, but it has the capability to do that, and certainly we get the understanding of each step, and we're able to propagate good practice across the various groups. So the platform, we'll start with a discussion with the people who are most familiar with the gene, determine the project aim and the route to transgenesis. And then I think a very important feature is that somebody is responsible for the design, and then there's somebody who is very dependent on getting the design right, checking it. So the design could be done within the team, the genomic manipulation service team, or it could be done by the lab. But there's that check to make sure we get it right. And we've got a couple of examples where things have been picked up which just wouldn't have succeeded in generating mice. Then we have included at the LRI an in vitro and ex vivo testing step by injecting into blastocyst and then doing PCR, because we wanted to assure ourselves of the possibility of firstly cut and then repair. And then finally, with the knowledge that that sequence is going to work, and most of them seem to, we can then inject, or indeed we can be looking to transfect, because there are certain projects that might still need to follow the cell route. And then the collaboration really has to carry on with screening, which, for the reasons already stated, can be really rather complex in comparison to a gene targeting or a pronuclear. And this is where we've got so far. And I should add that Natalia Monkow, who's on this panel, has really been an integral part of carrying forward the CRISPR-Cas effort on the part of the LRI. So we took on an initial tranche of projects around a year ago, mostly incorporating the design. We're now seeing a second tranche of projects where more of the design is handled in the lab. Most projects have been successful in terms of blastocyst PCR and at good rate, so that shows the design is good, and shows Natalia is an expert and should be sitting on the panel. So what we haven't, and this is now followed through, and we've generated mice. We've got various project types: conditional knockout projects, creation of indels, straight knockout, and point mutations, and all to various degrees have been successful. We've had a problem with low birth rates in mice, and we're now injecting at lower birth rates, and that's ongoing. And then, as you've seen from James's talk earlier, there's a big effort using CRISPR-Cas at NIMR, and other groups also involved, and success with those injections too. And at that point, I'll hand over to Sarah.
So yeah, I think Ian's shown that we have pretty successful CRISPR services working at the respective institutes at the moment, and we've got transgenic teams that encompass a whole array of facilities and services that we offer to everyone. What we're hoping is to give you all of that once we get to the CRIC, but also try and offer more, and we're really happy to receive any input on that. So rather than currently with some of the things that we offer, say at the NIMR, we offer microinjection. We've started to try and incorporate design into that, but we'll offer a service where you can come to us to design your mouse, and we'll pass over a mouse that's ready screened at the end, or you can cherry pick the services and come in at any point. So we'll try and kind of expand in that sense. We've got quite a large team, and we've got very different skills within that team. So hopefully that will allow us to kind of flex as things change. So with things like traditional methods of gene targeting changing and CRISPR coming up, we can adapt to those needs. Hopefully we can meet all species. So you've already heard about what we're doing with the opossums, which has been a very exciting challenge. We have always traditionally worked with rodents in both teams, and we've done a little bit of work with aquatic species, mostly fish. So we're hoping to expand a lot of the services that we offer to fish users and also to frog users in the future. And there's a couple of things that might work for fly users as well. And then some of the things that we offer, if they don't have those services at the other CRIC funding partners, that hopefully we can offer the same services to them as well. So this is just a quick overview of kind of what we're offering. There'll be a really big gene targeting and stem cell culture specialism, designing gene targeting constructs if they're still needed, derivation and characterization of stem cells and IPS, and then also just providing people with wild type lines, MEF production, transfection, and also karyotyping lines as you want them. And actually we're also moving into human work, so I'll be working with the Human Stem Cell Facility and offering all of those services for human stem cells and for human IPS cells. So we will be going to visit Bill later to get advice as to how we can offer that as a service. And hopefully we'll be able to offer training in stem cell culture as well. So what will be quite new to us is kind of beefing up the molecular biology in the team, helping with design of and advice of constructs. We already do genotyping, so we'll still be offering that. All of the wild type lines, both fish and mice actually, we're going to have a genetic stability program so that there's no drift in future, and we can stop genetic contamination. So we're sequencing all the lines, and they're available. And we're also looking at the microbiome health status of all of our lines at the CRICs. So we've started a project to sequence the 16S of all of our mice, fish, frogs, and we're going to do the flies. So you've got a historic look at that, and then into the future as well. There'll still be the microinjection services there, and also all of the cryopreservation, assisted reproduction, and importation of lines and rederivation. And actually that's kind of what both teams are heavily focused on at the moment, rederiving everybody's lines into the CRIC, working with the 1300 mouse lines, and we're also helping with the fish as well. So that's what we have to offer for the genetic manipulation service. And if anyone's got anything else they would like us to do, then just ask. So what we're going to do now is open the floor to discussion, really. We've got 5 experts. They're going to introduce themselves, give a little bit of a background as to their area of expertise, and then hopefully we can have a fruitful discussion. Lydia, would you mind?
Okay. Yeah, so my name is Lydia Teboul. I work at the Bill Gates Research Council in Harwell. In Harwell, we really have 2 ways of of looking into, we only do mouse genetics to start with. And we have two ways of using the mouse. One which is quite traditional, it's a little bit like what you were presenting, which is services which are bespoke. You know, people come in and ask us for specific mutants, and we make them for them. And we also have all sorts of phenotyping techniques to also characterize them further and cryopreserve them and distribute them. And the other thing that we do, which is slightly different, which is high throughput programs where we are producing a very large number of mutants which are made under the same scheme with the same characteristic in terms of designs. And these mutants are also systematically phenotyped, and all that data is put in on databases and publicly available. It's under the IMPC program. And indeed, what we are trying to do at this point, putting in CRISPR in the mix, is applying CRISPR in both these aspects of mouse engineering. Of course, to try to generate quickly and efficiently point mutation for the community, which seems to be a great demand. And also trying to look into applying the CRISPR tools to generate also as fast and as cheap as possible large number of lines which are then going to be able to be proposed and disseminated to the community.
Okay, my name is Nitzan Gonen. I'm a postdoc in Robin Lovell Batch Lab in the NAMR. I've been doing several things with CRISPR. So first of all, I've been deleting an enhancer of SOX9, and I did that in ESLs. This enhancer was 3.2 kb in size, and I did that using two CRISPRs, screened and did southern blot in ESLs, and then procedural services were generating the mice for me. So this is one thing I've been doing. Another thing I've been doing with a master student that we had in the lab was to generate a reporter line. So we had a gene which is called BTC, which is a secreted molecule, therefore it's very hard to detect it by immunos. So we generated reporter line by doing homologous recombination, again in ESLs. We removed the stop codon of the BTC and inserted a 2A TD tomato cassette with neocassette inside to select. And we also verified that by southern blot and generated, we're in the process of generating mice from that. And the last thing that we have been trying to do without much success, but I don't think it doesn't work, is to try to insert LOXP sites in order to generate conditional allele. So we did that again in ESLs using oligos. We tried single strand and double strand oligos in combination with CRISPR. And that's it, and I can answer questions regarding the design and the strategy to use, especially with the reporter gene, because all of the things that you're doing that will most likely result in you having a heterozygous line, you need to consider that you also have the other allele, and you will have probably indel mutations in that, so you need to take that into consideration when designing where to target your CRISPR, and that's all.
Hi, I'm Brendan Doe. I manage the mouse production for the Sanger Centre. So similar to Lydia, we are involved heavily in the IKMC, IMPC international consortia, where the aim is to introduce a mutation into each of the 20,000 known protein coding genes and put all of those animals through a primary phenotyping screen and make that data publicly available, and those mice publicly available through EMMA repositories and COMP repositories and so on. And so, of course, until now, we've been doing that with the S cells. We're now switching heavily into CRISPR, and we're confident that we can deliver exon deletions and point mutations, and we're now trying to work on using CRISPR to deliver more complex alleles, and Katharina can perhaps say a bit about that later. And so my expertise really is in mouse production. I'm surrounded by molecular biologists, so I'll let those people answer those questions. I can answer technical questions about the techniques itself, and if it's of interest, I've got some videos which I can show of the different ways to deliver the CRISPR-Cas9 into the mouse zygote.
Hello, my name is Katrina. I'm working together with Brandon. I'm actually based in Alan Bradley's lab, also at the Sanger in Cambridge. And my basically to-do list is optimizing CRISPR in mouse embryos. So I'm testing everything from making indels, large deletions, homologous recombination, basically trying to gauge how much is possible and what's the limit to CRISPR just in mouse embryos. That's basically it.
Hi everyone, I'm Natalia. I work at the transgenic facility at the LRI with Ian Roswell. And well, since last year that I started, and the idea was to set up the CRISPR facility. And the idea, as Ian told you, is from the design of the construct and the strategy, considering all the possibilities regarding the screening. That is the most, I think, most important part and not trivial at all, until to produce the proper animal and give it to the user. So we are kind of involved, we are a small team, but we are involved in the different parts, and we do all kind of injections and the blastocyst test that Ian told you. But basically, what we want to provide exactly is how to design what is the best strategy. If you go through the normal CRISPR injections, to go through the ESL targeting, so to work together with the users on how to design this.
So we've got quite a large block of time. So what we thought would be best is maybe if we stuck with certain subject areas. So we thought we could start with design and then move on to sort of delivery and screening. But it depends what questions people have. So has anyone got anything to start us off?
So in my personal case, I'm interested in deleting long non-coding RNAs. So because these are non-coding genes, it's not enough to just use one CRISPR and hope for a frame shift mutation or something like that. So what I need to do is to delete the whole lincRNA gene, or at least most of it. So what is feasible in terms of size? How big can these lincRNAs be in my case? So how many KB can we delete?
Are you talking in mice or cells?
So that's another issue. So I could either do this in ES cells to start with, or I could, I thought of another strategy, which would be to inject directly into mouse zygotes. But I'm guessing because I have to use at least 2 different CRISPRs that it's less efficient than using one CRISPR as one would use for coding genes.
Well, so there are several papers out there, and I have seen it quite often in cells. You can get 1 million bases easily, quite efficiently as well. So we have been doing that in the labs. In mice, the largest one I have personally seen is 150,000 base pairs. But I'm just waiting for pups to be born for even larger things, and I wouldn't be surprised if they work.
Okay.
So just try.
Was that through direct injection into
In mice, I designed 2 guides left, 2 guides right, and have an oligo that's basically stitching it together. So homologies, that's 150,000 base pairs apart, and you just pull it together, and that's working surprisingly well.
Injection as well.
And what about Mosaic? Have you checked that?
Well, they are just running on the MyStick, so I don't know.
Okay.
Thank you.
Can I just add something? Because I'm actually also working with a non-coding RNA. I'm also working with a non-coding RNA, and so actually we wanted also to create a deletion to silence the gene. And actually I've tried to remove the putative promoter. So sometimes it's true that you don't really know where is your promoter. But but we have designed only a couple of Cas9 guides, so 2 Cas9 guides, to remove more or less 1 kb of the promoter. I have tested the construction in fibroblast, so because we are working in opossum, so we are in fibroblast. And it seems to work quite well because I have managed, I have maybe, I don't know, 50% of efficiency. And then actually to check if your deletion is enough to silence your gene, you just have to do something like RNA-FISH or something like that to, it is gonna work, yeah. And then you can check. But I don't know, it seems to be quite efficient, but I remove only 1 kb because removing a very large part may work, but yeah, efficiency may be smaller.
Sorry, in your case, are you using repair template?
No.
Because I also heard that you can do it, maybe not that big, without using repair template, just cutting.
Yeah, you could, yeah.
Yeah, and I think you said something very important, which is double check that that you indeed lose the expression of your non-coding RNA, because with removing a promoter, it also can happen that you then reveal some promoter that were not used before.
But yeah, then you check if you have silenced your gene. And
Any other questions?
Hello. I'm trying to knock GFP into the C-terminus of my gene of interest, and I'm just having trouble making the homologous recombination donor vector. So I wondered what technologies people are using to insert GFP into their genomic DNA in order to make a plasmid to use as the recombination template.
So I've done something similar. I got a backbone from a friend which contained, so first of all the question is where, if you want to insert the GFP with the T2A or iris in between, or you want to have
direct fusion to the protein.
Direct fusion. So that can also work. I mean, I got a backbone from a friend that had 2 restriction sites to insert the 5' arm and the 3' arm. And then you have to decide what is the size of the arms that you want. I used 3 kb because I think it's big enough to have good specificity. But I guess you can do smaller than that. And then you need to make sure that you have that everything is in frame. And you need to consider that your 5' arm should have everything upstream to your gene just without the stop codon, and then to have in frame the GFP that you want. The 3' arm is another consideration. So if you want to, in your case it may be a bit more tricky, because if you want to put it in the 3' side fused to the protein, it may be more problematic because your chances of getting homologous recombination are good, but you're almost 100% likely to get one allele that has the homologous recombination. And the other one, because you have a CRISPR that mediates everything, you will have an indel mutation. And then your indel mutation, if you're doing it straight after the stop codon, your indel mutation will be in the stop codon, which will be very problematic to carry it over, because then you won't have...
I'm doing it in flies so I can breed it out, so it shouldn't be a problem.
Okay.
It's not a cell line.
Okay, so if you can breed out that, so I don't see any problem. I mean, most of the tricky parts in the design is to make sure what are you doing with the other allele. But can it be that, I mean, will it be enough in terms of expression if you'll have 50% expression of your gene?
My problem is cloning the vector at the moment. I've not even got to that point. But yes, it would be enough.
Okay.
It'd be fine. It shouldn't compromise the function of the protein. We've made this fusion before, and it doesn't compromise the function of the protein. I just want to do it endogenously.
Okay, so what are exactly the problems that you have with the cloning?
I've been trying different seamless cloning technologies, and I can't get any of them to work for my gene. I just wondered if anyone had had any success. I wanted to recommend a method.
Sorry, one thing I would recommend is the G-blocks from IDT. They work quite nicely. So you you have to, instead of buying single-stranded DNA, you buy blocks of double-strand DNA. They are quite cheap compared to the size of the fragment. They go until 4 kbs. And you clone in any vector that you want. You buy them with overhangs, or you make the overhangs yourself, you cut them, and then you clone them.
Okay, thank you.
I mean, and according to them, they also have a way that you can buy several of them and paste to each other. And they recommend always to use as normal traditional cutting and paste instead of gypsum assembly, because they always say that gypsum assembly is nice when it works, but there are a lot of situations that it doesn't work. So
just to mention something to the Gibson, I mean, we are doing that on a regular basis, and we are using the gene blocks. So basically what we do, we have a vector backbone, which we just PCR amplify, and then we buy gene blocks for the homology arms and have one gene block for the GFP or whatever. And so far I haven't had a single one failing. So you have to know what you're doing with your Gibson assembly. But once you know what you're doing.
Do you buy a kit for Gibson assembly, or do you have all of the reagents?
No, we're buying it in, yeah.
The NEB one?
The NEB one, yeah.
Okay.
And we have similar experience. We had some trouble at the beginning because we were trying to have homology region. We were too long, and somehow we got all sorts of funny things happening. But we are just shortening the homology, the end homology for the enabling the Gibson to happen. That really solved our problem. Probably, between 30, 40, that sort of size.
This is a technical question about sort of doing CRISPR in cell lines, or maybe I was just wondering if there's anybody that has any experience using sort of non-homologous end joining inhibitors to try and sort of drive sort of HR. I mean, I was trying to sort of create a point mutation, and I had a lot more success getting knockouts than actually getting, I didn't get any point mutations in there, so I was just wondering if anybody else had done that.
We're just starting, so I can't tell you.
Yes, we did just the first test with the SCR7. So far, we did the test injecting all the CRISPR system in the zygote, and we analyzed blastocysts. I have only preliminary data, and it didn't look very promising. We test only 2 different concentrations of that drug, and we did Surveyor. That it was not very clean. So we are doing it, definitely. And it has to be done, hopefully. I have other people from Germany that tried, I think, quite in a comprehensive study, and they didn't see any difference.
And we have,
I don't know.
The first thing we're seeing is at least it doesn't kill the embryos.
Yeah, exactly.
So it doesn't hurt, but we haven't seen magical enhancement with trying to put point mutation in.
Yeah. And has anyone else had any experience of that in the audience, working with the inhibitors?
We haven't had any experience, though.
Maybe we need to.
We've obviously not tried it, but a pal of mine at Cornell, John Scamenti, has used SCR7 with great success on pronuclear stage embryos, and he says actually the survival rate is better in SCR7 treated embryos. I think they do the injections, they incubate them in SCR7 overnight, and then they transplant them into recipients. And in his hands, the HDR, so the homologous recombination events are increased by about 10-fold. So they're now using SCR7 for all of their experiments.
Do you know whether it was for making point mutation or with oligonucleotide or with large donors?
This was actually for donor, using single-stranded donor sequences, but I think they were quite small.
Okay.
So maybe it's, that would influence there.
Hi. Do you think it will ever become feasible to do either gene knockout or mutation in the several copies of a gene in a cancer polyploid cell line, or is that just a non-starter?
I'm not too sure. So sorry, I was writing. Can you say it again? So
will it be possible to target genes in a polyploid cancer cell line?
don't have the option of going forward.
What do you want
Would
to
it-
do with them? To delete them?
But also, probably more likely insert a mutation.
I think it's going to be a game of numbers, and
Yeah.
the great news with the cell line is that you can have as many clones as you want, or as you can be asked to manipulate. So
just screen. Yeah.
yeah, and screen again. What I mean by that is you could start to affect a first population and then reapply your
Right.
reagent to go a second round if necessary.
About the second round, I wouldn't reapply with the same CRISPR because you're most likely damaged the sequence a bit, so you should use for this. So if you can check that you damaged, let's say, 1 or 2 alleles, and then establish a line from that, and then target it again with a different CRISPR, and then you know that you're
Okay.
minimizing it.
Can I get people's opinion on guide length? So I think the length of the guide RNA. So I think I'm right in saying that we're now routinely using 70 mers without seeing any obvious decrease in efficiency, with the idea it should reduce any off-target effects. And I just wonder if anyone else has experience with different length guide RNAs. What are people using?
20?
20 base pairs.
Is that because you saw off-target effects with 20?
No, we've never seen, but we haven't looked closely either. But we've never seen off-target effects. But it just seems a sensible precaution.
We have to say we've been screening our deletions for off-targets, and we haven't seen off-targets so far. So it seems to be in our hands so far, a scare that isn't coming through in the animals, as far as we can see. Obviously, we're not screening for every single off-target site, but we're screening for the most likely.
I mean, I was at a discussion recently where several people were using 18, I think, and finding that quite beneficial, but I hadn't heard of 17, but yeah.
Hey, hello. My question is just quite basic. And can you use a CRISPR-Cas technology for primary cell cultures, like bone marrow culture? Do you think that could be efficient, or you need cells that actually replicate in order to be able to do the selection and then get your alterations at your genes?
I think that the main problem there will be the efficiency of the transfection of that type of cells. No?
So I think the question is, first of all, for how long you can maintain them, and how well they proliferate, and how well you can transfect them. And regarding the selection, you can do the selection either with promycin, or there are plasmids available today with GFP, so if they can survive sorting, you can try to sort them. And if all of that I mean, if they live long enough, a few weeks, so I think there shouldn't be any problem with that, with doing.
Do you know of any examples in literature? Sorry, I don't know the literature that well in order to be able to say.
I don't know so many people who do that with primary cells, but usually it's because people want to get to an animal with that, or they're doing that in ESLs or straight in the animal, or if they're working with organoids, or for example, so they want to have a line to ask many questions. But I don't think that it should prevent you from seeing what you want to see in a primary cell. It just means that you're going to have to repeat doing that all of the time.
So just following up on that, there are a few examples in the literature with bone marrow, but you need to use lentiviral system because, yeah, as it was mentioned, the problem is the transfection. Yeah, they're not really transfectable.
Hi. So the lab that I just started working in, the model system is yeast, and I am just wondering if I would like to epitope tag my gene of interest in yeast. Is there expertise on CRISPR-Cas in this system? It's at the LRI that I've joined.
Actually, I had a question for Brendan. How are you finding injecting the protein Cas9 into the embryos? Did you have
in terms of what, efficiency, or in terms of
how you're
ease to
getting it
inject
in,
it?
ease to inject?
It's no different to injecting mRNA, so it goes in
just as well.
Just as well.
Okay.
So do you think it's better to inject the Cas9 protein instead of the Cas9 RNA regarding the mosaicism rate, for example?
We don't know. That's currently also on the MySeq. From efficiency, just the first few data sets I've gotten back, it's not as efficient as the RNA.
So is it a story of quantity of protein that you can actually inject?
Well, obviously I didn't go very, very high, so we could try that, but I thought I'd try equimolar comparisons. Obviously, if the mosaicism is lower with the protein, it might still be worthwhile because you don't have to breed that many. So.
And so do you buy the protein or do you purify
yes,
it
yeah,
yourself?
yeah, I bought them.
Because I have the microphone, can I continue? This is a very a technical question, but what kit do you use to generate your mRNA? Because right now in the lab we're using the one from Ambion, that is the mega short script T7, and sometimes the reproducibility is not good at all. So do you have the same problem, or it's just
so for the mRNA, the Cas9?
Yeah, both.
The mega machine.
Not the Cas9. I'm sorry,
guide.
the other one, the guide. Yeah. So sometimes we do it for a guide, and the rate is very good, and the day after we're going to do it again, and the rate will be not good.
I mean, I've seen the same thing. I think it's just basic handling problems. You had a bad
template,
day,
the quality of the template.
clean.
With one person doing it, maybe you could say that someone was having a bad day, but if you see it across like 3 or 4 people, and it's reproducibly irreproducible, which sounds stupid, but it is. We see like 800 nanogram per microliter, then the next day exactly the same conditions, same person, 100 nanogram per microliter. If someone else tries the guide RNA, they get the same thing, they get the variability. And we've concluded that maybe it's sequence specific.
So
like some guides don't like being synthesized by this enzyme.
Do you have your guides, I mean, as a duplex inside the vector, or you amplified, or you do the transcription using a PCR product with a T-cell?
PCR product.
Because what I do is I have all my guides in a plasmid that has a T7 promoter and the transactivator Cas9. And I mean, it's variable, but not as variable. I mean, I don't know
what kind
if
of
it
variation
helps.
do you see? Not tenfold.
No.
Okay.
No, definitely not. So we have a vector we linearize and we transcribe with T7 that is inside the vector. And maybe that helps for the transcription. I can give you the vector if you want.
Okay, cool. Does anyone else use any other kits for IVT?
Also from NEB, like just a T7 in vitro transcription kit, and that worked as well, and it's actually cheaper. So maybe you could also try other in vitro transcription kits. Yeah.
I have another technical question. Do you use the plasmid? I think the name is PX451, but I'm not sure. It's the one with the Cas9 with, I think, a 2TA and the puromycin. Do you use it, and do you think it's efficient? So me, I use it, for example, and my feeling is that, so I do my transfection, and when I put my puromycin selection, even if my transfection rate is very good, usually I kill almost everything. So my feeling is that the puromycin expression is not so good. So
there was a discussion about that on the CRISPR forum about the puromycin that has some SNPs that was resulting in not very efficient expression of the promethazine resistance. Check about that, because I think they make an update in that gene repository with a new plasmid.
Okay, good to know.
Hi. What do you think would be the best strategy to screen for both alleles? Would you always do topo cloning and cloning, or is there something more fancy and more high throughput that could be used as a way to screen both alleles after CRISPR knockout?
It depends on what is the mutation that you're after. Just looking at the melting curve is sufficient sometimes to see whether you are...
No, but I want to know the sequence read, not if there is or not cutting. I want to know exactly what is allele 1 and what is allele 2. What is the best way to do it?
Then you go to sequence it.
Yeah, but even sequencing, if you just sequence, you will get a mixed result because both alleles are different.
You can send it to MySeq.
So MySeq would be
Expensive, but yeah.
no. But so and for MySeq, would you PCR? I don't know about MySeq, but would you need to do a PCR around the area, and then send the amplicon for sequencing?
Yeah, so you PCR it, and then you add adapters that are basically your barcodes that you can sequence more than one sample at once, and then you just send it, and then you get back a lot of files that tell you how many percentage of whatever read you have in there.
So meaning that if you do barcodes, can you run more than one sample at the same time?
Yeah, I'm running 96 at the same time, and there is one setup for 384.
So basically you make some forward primers with barcodes, reverse primers with barcodes, and then you just pull everything and you send for sequencing.
Yeah, similar, yeah.
So that would be the best way. Okay.
Yeah.
Thank you.
I'm just wondering, are we going to have a website or something where all this information is at the end of the day, so that we don't have to sort of rely on memories and scratch notes?
Christoph, do you want to answer that? I think you've set up.
Yeah, the idea would be to maybe have this on the Creek net, at least at the beginning. So I know it will be for Creek people, but then I will see if people are interested to have the information I can distribute.
Oh yeah, didn't you mention that we are being taped? Yes.
I have a question about when you co-inject. So when you co-inject in embryo the Cas9 RNA and something, a piece of DNA to do homologous recombination. So what do you do usually? Do you do pronuclear injection in the nucleus, or do you do it in the cytoplasm? My other question is, if you do it in the cytoplasm, how does your piece of DNA go into the nucleus? So how does it work? Because there is no nuclear localization signal. So so I just would like to know if you, well, so yeah. So do you do pronuclear injection in the nucleus?
I think a lot depends on one, what skill set you already have within your team, because a lot of teams already have the ability to do pronuclear injection, and you can see quite easily what you've injected. Another question, of course, is what allele you want to make, and so if you want to make an allele with a more complex vector, targeting vector, a large targeting vector, I think possibly you'll need to go pronucleus. For things like SNPs and point mutations, we've been very successful at making those just with cytoplasmic injection. And so I guess the advantage with the cytoplasmic injection is that you're not potentially damaging any chromosomes when you do the injection that you potentially could be when you're doing a pronuclear injection. And they've been successful using that method.
And so do you always inject, so when you do cytoplasmic injection, do you always inject pronuclear stage embryo?
Yes.
Meaning that you don't have the envelope. Because we are working with Oposome and collecting pronuclear injection, pronuclear embryo is almost impossible. So do you think if we inject one cell embryo, do you think we will automatically have mosaicism because we will have to wait for one cell division to have the plasmid going in the nucleus?
I'm not sure.
Because otherwise, how does your plasmid can go to the nucleus? Do you think the Cas9 guide, I don't know, the Cas9 take everything and bring everything to the nucleus?
Possibly, but also you remember the pronucleus stage, as the day goes on, breaks down and you lose your pronucleus. And so the Cas and the guide is still active.
Yeah, but if you take a one-cell embryo already.
But the one cell has a pronucleus.
Yeah.
The one cell stage embryo has a two pronucleus. That's a stage that you need.
Okay.
And then when the membranes of the pronucleus, both pronucleus start to break.
Okay.
And then that's what your DNA is around there.
Okay.
And
I can show a video if you want.
yeah, that would be nice.
Yeah.
I mean, Sophie, correct me if I'm wrong, but I think we found that if you only want to target one of the alleles, we get slightly better. We'll go for pronuclear, won't we? Whereas you get. Yeah.
I think we had difficulty getting. Sorry. I think we had difficulty getting both alleles when we were injecting into the pronucleus.
Yeah. So if you wanted to just target one, and there are occasions when you do, it's
Yeah,
better to go pronuclear.
but it's as Brendan said, you cause much more damage when you inject the pronucleus. I mean, I'm transferring 15 one-cell embryos per female to get the same results as I would normally transfer 20 for a pronuclear injection when I inject into the cytoplasm. So they recover so much better, you put fewer in. So.
Yeah, I'd agree with that. I mean, doing cytoplasmic injection versus pronuclear injection, the recovery of pups after transfer is much greater with cytoplasmic injection. And so I think traditionally doing a typical pronuclear injection, we would have cultured the embryos to 2-cell because of the damage that would have happened. But with cytoplasmic injection, if you culture through to 2-cell, virtually everything that survives the injection divides through to 2-cell. So we tend to replace it 1-cell now. So this is a cytoplasmic injection. And so you've got your RNA in here. These are the pronucleus just up here, which we're avoiding with this. And so in a mouse embryo, the eulemma, the membrane here, is extremely flexible and it's actually quite difficult to break. And so what you find with the injection is you have to pass the injection needle all the way through, right to the other side before you break the membrane, and then you see a rapid release of the RNA. So you can see the RNA coming out. And this is using a femtojet, so this is a positive pressure injection. There's a continual flow. And what you're looking for here is that kind of whoosh there, and that's an injection. So the difficulty is, though, that most teams don't have cytoplasmic injections as part of their toolkit. So there is a training element to this. But we're now getting routinely 80–90% survival with cytoplasmic injection within the team. So it does work well.
You touch on a very important point, which is a matter of skills. It depends on how much time you got to invest into honing this new skill. We decided to take the different tack, which is to keep with pronuclear injection, but we also get a very, very high level of efficiency. Probably 80–90% of the pups that are born have mutations. Yeah. So the fact that we lose some pups in the way is not something that we find that difficult to live with, because then you find yourself with F0 that you have to breed anyway. You don't need to have lots of F0s, you just need to have a handful.
Yeah, no, I'd agree with that. I guess the advantage for us is that we've been able to reduce quite dramatically the number of eggs we inject per target now. So we're down to 50 to 60 eggs per target, and we're confident we'll get a deletion injecting that number.
And we are on similar numbers in that respect.
Yeah.
Are there any more questions? Anyone? James?
I had a question for the non-NIMR people on the panel, which might be of relevance to other members of the audience. What sort of experience have you had with injecting different strains of mice and survival of different strains?
So we necessarily, for most of the projects we're involved in, have to inject C57 Black 6N for part of the IKMC, IMPC pipelines, where it has to be on a standard background. So most of the injections have been done on that. But we have done injections into F2 zygotes, and the feeling is that they're a bit more robust. They withstand injections a bit better, survival rates a bit better, and the birth rates are a bit better. But Black 6 is working perfectly well, actually. The numbers are pretty good. We're getting 30% birth rates from Black 6N doing cytoplasmic injection, which I think is not too bad.
Similar experience at Harwell with Black 6N and Black 6J. The one that we haven't managed to get success with is C3H. Just we haven't been able to get any founder out of that. But the inbred Black 6N, Black 6J is working. We only do those. We don't do hybrid.
So C3H, the survival rate of the injection is okay, or is
Yeah,
it just
we had a survival rate that was decent, but nothing was mutated.
So, sorry, just to put that into practical terms, I know you might have the numbers at hand, but what sort of numbers of pure B6s would you have to inject to get mutation rates as high as your F2s?
Well, I mean, standardly we're injecting 50 to 60 Black 6N embryos, and most of the time, I think currently we're at about 85% of those projects give us first time round a mosaic F0 with the correct allele present. So, yeah.
So still about injection, do you use now the commercial Cas9 RNA, or do you still do your own Cas9 mRNA?
I myself use the TriLink one, the commercial one.
Okay.
Well, as I'm testing all, I'm trying all, but the TriLink is working very well. So if you're just trying to make a few, then I would buy it.
Can I ask about what type of indels have you been seeing? Are they small? Are they big? Are they really big? Are they really small? Is there any trend?
So from what I've seen, you see everything from even SNPs to, I mean, the problem is some of them you don't pick up because your PCR is at distinct lengths, but I have seen 250 to 300 base pair deletions in the MySeq data, so you get everything.
And large insertions too.
Yeah, large insertions too, yeah.
150, 200.
Yeah, 100 I've seen at least, maybe a bit larger than that, yeah.
When I was sequencing my deletions, my indels on the undeleted allele, I could see only deletions. I didn't see any insertion, and the deletions varied from 2 base pair up to 20, 30, but that's not as big statistics as they have.
There seems to be also a tendency, depending on your design. Some designs are prone to make deletions. Some designs are prone to trigger insertions. It doesn't seem to be completely random. And if you're trying to do a point mutation with an oligonucleotide, you sometimes have both a good footprint of your oligonucleotide and just associated next to it an indel. So it's not one or the other, it can be both. And it often is both.
Following on from the last question, when you're screening for mutations, if you're looking for just a straight knockout animal, are you selecting for those that are smaller and kind of frame shift, or do people have a preference as to what size mutant they'll carry forward?
It's completely project specific. Yeah.
So if you as project specific, as in for the gene.
Yeah.
So if you just want to disrupt the gene function.
If you want to disrupt the gene function, you try to aim for a frame shift. You also try to avoid deleting blocks that would be potentially conserved, for example, that are nothing to do with the coding sequence that you're trying to affect. And so the more you know about your genes and what are the important blocks, not only in terms of expression, the more you're able to make the call of which one you want to carry forward.
Has anyone got anything else they want to talk about? Has anyone on the panel got anything they want to bring up?
I just want to mention that another option, even though it's becoming less attractive nowadays, is to do tetraploid complementation assay that we have been doing with Sophie. So if you have something, if you're generating what you want to generate in ESLs, so you can, and you verify everything by sequence or Southern blot or whatever you want, and you have it homozygous, you can inject it as a tetraploid complementation and get within 3 weeks your embryo with the right phenotype without needing to breed anything. But there are also limitations to that, but it's another option to consider.
And maybe it's a much better option than considering to phenotype your F0. So the product of the pronuclear injection or the cytoplasmic injection, even if they look non-mosaic at the point of genotyping, we have had lots of good and bad surprises in terms of what is being transmitted. So a clean sequence at the ear biopsy doesn't give you the whole story of what is going to be transmitted by the founder.
So we do have a tea break scheduled now, but I just wonder whether there's still a lot of things that people want to discuss or not, and whether it's worth carrying on a little bit now and finishing early, or whether people have enough to come back later.
Does the panel want to discuss off-target effect and how to screen them?
To screen them or not to screen for them is another way of asking that question. If we follow on from what I just suggested, is that the founder is never something that you want to phenotype or to study, then you find yourself in a situation where you are now breeding your line and in effect diluting any off-target effect that you would have put in inadvertently, unless they are really, really closely associated to what you want to affect, of course. So that, together with the papered experience that we have across the different people doing this, suggests to us that this is not really something that is going to be much more of a problem than if you are making a mutation in ESL and then getting a germline transmission from an ESL. ESL themselves are not perfect in terms of their genome, and we live with it by just at the beginning. A few years, many years back, people were asking for 2 or 3 clones being transmitted and separately assessed to actually say that the phenotype was really associated with your gene targeting. You could imagine doing the same thing. And the other thing that you could imagine doing is follow that very good advice that Bill mentioned earlier, which is trying to revert your point mutation. If you really want to be sure that what you're doing is what you're studying is linked to a point mutation that you would have put in.
Yeah, and I also think that working with cell lines and using CRISPR with cell lines is a completely different scenario than working with the zygotes that we are working on. So in the cell lines, we are transfecting with a plasmid that is a ridiculous amount of guide RNA that you will have there and Cas9 that will last longer, definitely, that the RNA you are injecting in the cell line, so in the zygote, sorry. So that's probably, all the reports on off-targets are mostly on cell lines. There's no report or significant reports on zygotes, on embryos that have off-target effects. I mean, and you've screening frequently, and there's no effects. But yes, on the different cell lines.
Do you think you could put a degradation signal on the RNA to sort of make the effects, I don't know, make it degrade quicker so you get less mosaicism?
If it's possible, it would be great. I don't know. Yeah, I have no idea. But
where do people think the mosaicism arises from? Does it arise from the cell division occurring, and then so 2 mutations happen in 2 cells, or does it happen in one cell, and then the protein hangs around or the RNA hangs around, and then it happens again and again because it's hanging around, it's not being degraded.
I think that's the theory.
So it's hanging around. It's not the fact that if you injected it earlier. So I guess in my head, people were doing the protein because the protein's in the ready form, so it will be used, and therefore you'll get one mutation and one mutation only. Whereas if you inject the RNA needs to be translated, and so you get the chance of it being divided. Whereas if both are hanging around and you're getting mosaicism because they're hanging around, surely if you degrade it quicker, then you're going to reduce your window, but you're also going to reduce the chance of mosaicism.
Yeah, that's the theory, but I think what does happen in practice is that when you microinject, you don't really have a strong handle of how much you microinject. So you are, because you can't calibrate that, you really never can calibrate exactly when and how long you're going to have your system active.
Has anyone made any Cas9 fusions to anything, and how did that go? No.
They are available in Adgene, so the Cas9 and Puro, or Cas9 with 2A and Puro, Cas9 2A with GFP. This is the plasmid that Fanny was talking about. So you can just order them.
We have just imported the Cas9 mouse, which will be available to everyone in the CRIX soon in the next few weeks. So if that's of interest to anyone.
You can also generate ES cells from that.
You can, which could be very useful. And we should.
Has any of you looked at integration of the Cas9 plasmid on the genome, or of the guide RNA plasmid on the genome, or at the oligo on the genome after transfection? If there is, I'm asking if there is a possibility of an extra layer of off-target by the reagents that you are using.
So when, at least when we are injecting into the zygote, you only inject mRNA.
Yeah. So what about cell
plasmid? But the template. Yeah. So when it is a single-stranded oligo, I mean, a single-stranded cannot be integrated.
Yeah.
And
so
when you use a plasmid, ideally you inject circular.
Yeah.
And
yeah. So what about people doing cell lines and plasmids? So does anyone has?
There is one report in literature, not with CRISPR, but with TALEN, where an oligonucleotide which was injected single-stranded ended up as a footprint somewhere else in the genome. Yeah. By the JAKs. Yes, there's one. It's a CRB1 mutant.
Okay, thanks.
So it's not a real concern.
But is there a real concern because people are not looking at? I think that is my worry.
People have been doing some whole genome sequencing with when they were trying to do mutagenesis with TALEN, and nothing has been seen. There's also people doing whole genome sequencing with CRISPR, and they don't see oligo, appearing here and there and everywhere. I don't think it happens much.
But it can happen when you make like cell lines, like we made like CRISPR knockout cell lines in HeLa and in HEX, and you use the GFP-Tec Cas9 because after transfection we then FACS sorted the GFP positive cells and then selected from those like single clones, and sometimes you can see also GFP positive cells in your clones, so it means that the plasmid for the Cas9 got integrated and that they are stable. Yeah.
Well, that's been my worry, because since you are sorting for GFP positive, or if you use Puro, you are sorting kind of enriching for the ones that are Puro resistant. Maybe you are selecting for some that actually got the stable integration of something that you didn't want to integrate.
Yeah, with the GFP-Tec Cas9 you can see it actually, or sometimes in clones it's also like variables that some cells have a strong GFP expression and some don't. So you could then in this case like maybe resort your clone and just FACS sort for those that are non-GFP. Then
so I have an extra layer.
Yeah.
So I did something similar in ES cells, that I did transfection with poromycin plasmid with a Cas, and then I wanted to do a second round of CRISPR to delete the other allele, and before I did that I checked for the cells that they are not resistant for poromycin, and all of the clones that I had 14 clones, and all of them were negative for poromycin. So don't think it so much integrates. But you need to check that if you want to retarget with the same plasmid.
Hi. Continuing with the off-target effects, what's the standard in the field to make sure that your phenotype is because of your mutation you intended to introduce? Is the idea of complementation with cDNA the best approach?
I think the best approach, both for you seeing that the phenotype you're seeing is coming from the mutation in your gene, and both in order to rule out off-target effects, is just to show the same phenotype with 2, 3, 4, 5 guides, and the chances of you seeing the same phenotype or having this phenotype appearing due to off-target effects when you're targeting different sequences is basically 0. So I think this is the best way.
We've done work with using 2 different guide RNAs, and we've realized, this is all cell lines, that the clones behave differently. I don't know if anyone has seen this in the room as well. And we're starting to wonder whether there are some compensatory changes in these cells when you grow them. So my feeling is that the only way we can confirm this is by doing a cDNA complementation to restore the phenotype. But I don't know what the others are thinking.
Did you check? I mean, I know that from Vivian's lab that they were working with cell lines. They had a problem of diluting the cells that have the phenotype, and it may be because when you picked it wasn't really a single cell or something, so it may be worth to do another round of picking and verifying that you're still with the same cell, and then it may appear. And regarding seeing different phenotypes with different guides, it may be an outcome of what you damaged in the protein, so it may have a different effect. This is what I think.
Thanks.
Because I've been doing making CRISPR knockouts, and I've actually been using, I've been creating sort of double knockouts, and these are, I've sort of picked multiple clones, and actually sort of 2 clones that I focused on showed me the right phenotypes that I was expecting, and then after I started doing more assays, I realized actually that they have subtle differences. And the only way around that was to just go back and pick more clones. And I think, and I know in this case, like they were specifically from single cells because I'd done sort of dilution in 96 well plates and, sort of started off from an actual single cell. So I think just you've got them under drug selection and you're putting all sorts of pressure on them. And so I think they must accumulate some kind of, and both of these were created using lentiCRISPR. So they had, the same guide RNA, it was integrated, it was probably, generating some kind of off targets at some rate. But yeah, multiple clones is really the only way to go. And you can never be 100% certain because, for some assays that I tested, they behaved identically, but it was in others that I actually saw that there is clearly a difference. And they were both P53 positive, so it's not that, one was growing better or worse because they had lost P53, but for whatever reason, in some growth assays, they behaved quite differently, and in others, they were just fine. So it's just complementation, multiple clones, different guide RNAs. It's a lot of work, but somebody's got to do it.
Sorry. And if I could add something to that, probably some of those phenotypes are just basically due to clonogenicity, because you start with a cell line that's basically a pool. It's a mix of clones, and then you do CRISPRs, and then you pick random clones that probably you start with a population that is not homogeneous. And now you are comparing something that is homogeneous to a population that is not homogeneous in the first place. So the experiment is screwed from the beginning. So I think what you have to do with the clones, you have to do with the parental, with the cell that you targeted. Then you might need to also go to a clonal step of that parental and then compare those clones with the clones that you get from CRISPRs. That's probably what I would do.
And every time you'll subclone, every time you create more diversity. So
exactly. So
just game of number again.
done. If no one's got any more questions, thank you to everyone on the panel. That was a really good discussion. There's tea served out the back, so we can have more sort of discussions in smaller groups back there afterwards now if we want. And Christoph, do you want to say anything? No? Well, can we just thank all of our speakers today? I think it's been a fantastic event, and thank you to Christoph for organising it.